Assalamualaikum and hello, I am Roharsyafinaz binti Roslan with metric number S51635. My final year project title is a review of cinnamic acid derivative as antimicrobial agent. Firstly, bauxite cinnamic acid is a class of aromatic carboxylic acid that occur in plant naturally. Cinnamic acid is the secondary metabolites. They are produced by a biochemical process that produces lignin. LCDC cinnamic acid can be synthesized with high yield and selective using different catalysts with a range of waste reduction and recovery procedure through Novenagel, Perkin, and Patchman reaction. Compound that will be focused on this review paper is para-coumaric acid, ferulic acid, and cinnamaldehyde. The antimicrobial activity have been determined by value of MIC. The MIC value was determined by against the gram-positive and gram-negative bacteria and fungus at the minimum concentration of solution. The antimicrobial activity of drug influenced by their structure and the physical-chemical properties. The objective for this review is to review the progress of cinnamic acid derivative and antimicrobial activity and to review the structure-activity relationship and physical-chemical properties of the compound. The para-coumaric acid caused the improvement in outer membrane permeability. Log P is a critical instrument for determining the hydrophilicity, lipophilicity of compound. Para-coumaric acid have positive value of log P, then it means coumaric acid increase the lipophilicity properties. Antibiotics, enthromycin and rifampin were chosen as probes to assess the improvement in outer membrane permeability caused by para-coumaric acid. The presence of para-coumaric acid, however, as dysentery become more responsive to the probe antibiotics at all concentration. The negative surface charge of para-coumaric acid can attack the outer membrane through electrostatic interaction and chelate ion magnesium 2+, destructing an OM and cause the barrier mechanism to be lost, the nutrient flow be blocked. Under electron microscopy, after 90 minutes of therapy, para-coumaric acid could trigger minor cytoplasmic leakage. 3 hours later, the membrane of para-coumaric acid treated cells started to burst and cellular content began to leak greatly. Membrane disruption. Next, the modification of cytosine ferulic acid caused the synergistic effect and increased the antimicrobial efficiency. Cytosine ferulic acid have been coated in the mangoes. Thermal coating can maintain the fruit quality and inhibiting fungal growth, which improve the appearance during transportation and storage. It happened because positively charged cytosine ferulic acid will attach to and permeate the negatively charged fungal cell membrane. This will result in increased the plasma membrane's permeability and leaking of intracellular component, ultimately resulting in fungal death. Thus, the CFA coating can significantly delay the yellowing process and increase the peel luster. And last but not least, cinnamaldehyde shown more effective against gram-positive bacteria. Cinnamaldehyde, as an antimicrobial agent with an aldehyde group in its structure combined with a highly electronegative carbon double bond, interferes with biological process requiring electron transfer. It covalently attached to a nitrogen-containing molecule such as DNA and protein through their amine group, thereby eliminating microbes. The source of article for this review paper is from Google Scholar, PubMed, ResearchGate, and Science Direct. The keyword have been used is cinnamic acid, coumaric acid, ferulic acid, cinnamaldehyde, and antimicrobial activity. The total articles that have been reviewed is 58 articles. The result shows log P is an important parameter because it shows the hydrophobicity or lipophilicity of the compound. This is since lipophilicity plays a significant role in a compound's absorption, distribution in the body, penetration through critical membranes and biological barriers, metabolism and excretion. The observation decrease in MIC value was closely related to an increased lipophilicity, but strategically position hydrogen bond donor group which promote interaction with membrane phosphorylate, as shown in the table. Ring substitution, hydroxyl and methoxyl group, and the saturation of the side chain, cinnamic acid, are all factors that influence the lipophilicity of phenolic acid. The electronegative of the double bond in the cinnamaldehyde much help in eliminating the microbes because it can covalently attach to the nitrogen-containing molecules such as DNA and proteins to their amine group. The hydroxyl group on the coumaric acid structure shows has no significant influence on an antifungal and antibacterial activity, but the presence of the methyl group in the ortho position on ferulic acid, the hydroxyl group may influence the inhibitory action. Last but not least, there are a few research shows that combination of two compounds such as cytosine and ferulic acid existing the synergistic effect that may increase the effectiveness of the antimicrobial activity at the lower MIC value.